Migraine is a widespread, crippling, and yet underappreciated and undiagnosed disease whose causes are not well understood. Therefore, the urgency for studies that could advance our understanding of its mechanisms has never been so high. Luckily, one such study recently presented itself. This research article compares the site expression profile of proteins that are implicated in migraine between rats and humans, as well as identifying sites of therapeutic action in humans and enhancing our understanding of the mechanisms of therapeutics. This study has the potential to determine whether rodents can be suitable models for further studies in the field of neuroscience in humans. So to begin with, what was the current state of knowledge surrounding migraine prior to this study? What are the proteins that are implicated in migraine? Well, it was known that inappropriate activation of a section of the brain tissue called the trigeminal ganglia is responsible. Once a TG is activated, CGRP and other neurotransmitters are released from neuron cells in the TG. And CGRP and other neurotransmitter proteins cause inflammation and vasodilation by binding to receptors on target cells, causing the migraine symptoms. Right, so what neurotransmitters and receptors did this study look at specifically then? One of the proteins studied was CGRP, which is a key neurotransmitter involved in migraine. As mentioned before, during a migraine, CGRP proteins are released from the fibers of neurons. These bind to CGRP receptors on blood vessel cells and those on other neurons and other cells. This in turn causes CAMP and calcium signaling cascades, leading to the migraine symptoms. So therefore, if we are to treat migraine, CGRP release from neurons must be minimized. PASAP, another neurotransmitter, and its receptor proteins, PAC1, VPAC1, and VPAC2, were also examined in this study. PASAP has high affinity for PAC1 G-coupled receptors found on blood vessels and other cells. These receptors promote calcium and CAMP signaling cascades, as shown on the bottom left, leading to migraine pain. So if you want to stop migraine here, you're going to have to antagonize these receptors, which means to deactivate them, essentially. Next are the 5-hydroxytryptan 1 receptors, specifically the 1B, 1D, and 1F subtypes. These receptors can have an inhibitory effect on neurotransmitter release. They induce signaling cascades that prevent the release of neurotransmitters such as CGRP into the synapse to target vessels and other cells. So if you want to stop migraine, you're going to have to agonize these receptors, and this means to activate them in order to block neurotransmitter release. Lasmitadine and the triptans, which are agonists, target these receptors. The final proteins analyzed in the study were SNAP2A and SV2A. These are involved in vesicle fusion to the membrane in neurons, allowing for release of CGRP and PASAP from them. SV2A, which is on the vesicles carrying the neurotransmitters, binds to SNAP2A and other proteins on the cell membrane, forming the snare complex, which permits vesicle fusion and exocytosis of these neurotransmitters. Botulinum toxin A, or Botox, can be used to target these proteins. Botox prevents vesicle fusion and release by cleaving the proteins in the snare complex. So if you want to stop migraine, Botox treatment is also needed. So that's plenty of background details, but what is the relevance of this study to migraine therapeutics? Well, as we've just seen, the neurotransmitters and receptors involved in migraine can be targeted to treat it. So comparing and mapping expression of these proteins between humans and rodents could identify regions of the TG for targeting, and it could also enhance our understanding of the mechanisms of action of therapeutics, therefore aiding in headache treatment. In addition, this is the only research so far that compares the expression of migraine proteins across 2 different species. This can potentially allow researchers to identify the translational value of studying neuropeptides in rats, right? If the expression results are similar, findings from rodent studies of neuropeptides could have some clinical value in humans, for instance. So how exactly was the TG of both species studied for expression, and what were the findings? Firstly, rat TG tissue was prepared from 6 rats and was dissected from 6 rats and prepared. Human TG was then also prepared via autopsy from 6 deceased adults. The tissues were then examined for expression of proteins via antibodies that are specific to the protein of interest, and that are also conjugated with a fluorophore. And this allows visualization of cells expressing the proteins of interest. This technique is known as immunohistochemistry. The results of the expression of expression from 5 proteins in the TG are displayed to the right. There are flaws in this approach, though, due to the phenomenon of autofluorescence. And this is basically just a natural body fluorescence from older people especially. Yeah, the phenomenon occurs in older people particularly. And this could create false differences in fluorescence between people between the TGs of people. So controls had to be used for each individual human studied, right? Right, so that's how it was generally done. So what are the findings of the expression patterns between species? So the table to the right here identifies whether or not a protein of interest was expressed in certain sites of the trigeminal ganglia. Expression of these proteins were found in the neuronal soma, which is basically the cytoplasm and nucleus of neurons, the satellite and the satellite glial cells, and these cells surround the neurons, by the way, the neuronal somas, by the way, and also the neuron fibers, right, which mediate the pain signals of the TG. And a positive expression result is indicated by an X to the table on the right here. What was found was that there was considerable overlap in the expression of all these migraine proteins between species, in particular cell sites. For example, PASAP were found in neuron somas and satellite glial cells for both species. Hence, there's obviously potential translational value for studies of neuroscience, such as those of studies of neuropeptide properties and migraine in rats. Right, rodents could potentially be used as model systems for clinical developments. So CGRP and in addition to this, CGRP and PASAP were both expressed in the same TG neural cell sites across rodents and humans. And this indicates there is a potential for anti-headache treatments to target both PASAP and CGRP release simultaneously. The potential for migraine therapeutics is clearly very strong. But what are the potential sites for all of the specific types of anti-headache drugs that were mentioned previously? So first, let's take a look at sites for antagonists of PASAP receptors, such as VPAC1 and PAC1, which prevent them from binding PASAP and causing migraine symptoms. The diagram to the right and the diagram depicted to the right here are the results of an expression co-localization experiments for neuron somas and satellite glial cells in rodent TG, and this also involved immunohistochemistry. The satellite glial cells surround the neuronal somas, by the way. The red colors here indicate locations where there was expression of PASAP, while the green colors indicate expression of the receptor target VPAC of anti-headache treatment. The yellow colors indicate locations where there was co-expression between PASAP and the receptor target. A key finding here was that VPAC1 was found to be expressed in satellite glial cells surrounding the neuron somas that were found to express PASAP. This means that drugs have the potential for inhibiting the interaction between the neuronal soma and the satellite glial cell. Therefore, hence SGD, satellite glial cells, should be a key site for anti-migraine treatment. But remember, the PAC1 receptor has a high affinity for PASAP. The results of this research revealed that this was expressed in neuronal somas only in humans and rodents, and this is not shown in the diagram to the right, by the way. So hence, the somas should also be a high priority site for PASAP therapeutics. So now, how about potential sites? So what about potential sites for 5-hydroxytryptan-1 therapeutics, such as triptans and lasmatadine? So remember, HD1 receptors reduce neurotransmitter release when activated. Triptans target 5 HD1 receptors and agonize them, reducing this release. The arrows in the immunohistochemistry results to the right indicate sites that showed expression of the protein of interest. What was found was that at least one of the subtypes were expressed in all 3 TG sites studied, but particularly in the somas and SGDs in humans and rodents. Hence, these results show that all 3 sites should be targets for the therapeutics, but especially the neuronal somas. However, there were differences in 5-HT expression between the humans and between the individual humans studied. So these results are not conclusive. With regards to the potential sites for these drugs, right? So perhaps more human subjects need to be tested to get a general understanding of the therapeutic sites of interest, right? The sites that express these receptors. Right, but how about targets for Botox therapeutics then? So remember, Botox degrades SV2A and SNAP25, which are crucial for the exocytosis, or the release of neurotransmitters. These results show that SNAP25 is expressed in most neuronal somas, while SV2A proteins were expressed in SGCs and some neurons in both rodents and humans. Therefore, satellite glial cells and neuron somas are important sites for Botox treatment to prevent neurotransmitter release. So in conclusion, to summarize, there is considerable overlap between the expression of these proteins between humans and rat trigeminal ganglia. And this could prove the value of rats as model organisms for researching neuropeptides and their mechanisms in humans. Numerous sites for anti-migraine drugs were also identified, which could lead to further understanding of their mechanisms of action. And in addition to this, very important sites for headache targets were also identified, such as sites where there was co-expression between PSAP and CGRP. But there are areas to explore further, right? How could the findings of this research be taken further then? So as mentioned previously, PSAP and CGRP were found to be co-expressed in TG cells. More studies are perhaps needed to determine whether PSAP and CGRP signal to other tissues in similar ways. This will help understand pain progression more. The TGs of a broader range of individuals should also be examined to see the main differences in migraine protein expression, in order to understand why certain anti-migraine therapeutics are not equally effective between people. There are also a number of different types of neuron fibers involved in pain progression. The researchers can investigate whether neurons expressing neurotransmitters, such as PSAP, are associated with particular fibers responsible for pain progression, to identify if these fibers are priority sites for therapeutic treatment. And finally, we can investigate whether these proteins, whether all of these migraine proteins are expressed in cell types other than neurons, in order to discover any further sites for therapeutic treatment. And that is the end of this talk. As you can see, this study has taken a small step forward in understanding migraine therapeutic mechanisms and the suitability of rodents for neuropeptide studies. More focused and specialist studies are needed to take our understanding further still.